Following our previous discussion of innate immunity, I also want to briefly introduce antigen-specific, or sometimes called adaptive immunity. These are some terms that we will talk about. Most importantly, we're talking about things like antigens, because antigens are the substances. Generally, they're going to be proteins that are recognized by the immune system. Another very important term is epitope. This is going to be the smallest molecular structure, again, generally smallest polypeptide that can be recognized by the immune response. Things like immunogens will elicit an immune response. It doesn't always have to be a protein. Sometimes various carbohydrates or even things like LPS from gram-negative bacteria can elicit an immune response. And then we have things that are T cell or T dependent antigens that have to be presented to T and B cells. B cells will lead to antigen production, of course. And then there's T independent antigens. Those are going to be some of your PAMPs, things that are just kind of broadly recognized, like bacterial structures, flagellin, lipopolysaccharide, and other various carbohydrates. The first type of cell belonging to the adaptive or antigen-specific immune response, of course, is the T cell. T cells are so named because they mature in the thymus. They are produced in the bone marrow. There are various types of T cells and T cell receptors. I am not going to get into all the details because, again, this is not an immunology course, but it is important that you understand the two major types of cells of the adaptive or antigen-specific immune response, T cells being one. In general, T cell activation occurs when the receptor on a T cell, and again, there are multiple types of T cells, when the receptor on a T cell comes into contact with an antigen that is presented by an antigen-presenting cell. This could be a macrophage, or very often is going to be a dendritic cell. When we do have binding of a T cell receptor to an antigen being presented by an antigen-presenting cell, we get this complex cascade signal transduction, eventually leading to transcription that can lead to things like cell proliferation, increased expression of cytokines, etc. There are two major groups of T cells that people talk about. They're called CD4 positive and CD8 positive. Sometimes people call them helper T cells and cytotoxic T cells. They each have different but very important functions. So with our CD4 positive T cells, or our helper T cells, they are presented antigen through MHC class 2. So MHC class 2 is found only on professional antigen-presenting cells, so things like macrophages or dendritic cells. And the idea here is that these help drive other immune responses to help eliminate infection. CD8 positive T cells are activated through binding of MHC class 1. And MHC class 1 is found on all cells that have a nucleus. CD8 positive T cells are also called cytotoxic T cells or killer T cells, and that's because when they are activated, they can trigger a target cell to undergo apoptosis. And the reason for this is because presentation through MHC class 1 most often means that there's an intracellular issue. We are in a medical microbiology class, so we're talking about an intracellular infection, most often a viral infection. But we could also be talking about something like transformation into a cancerous cell, which leads to the presentation of altered antigens that don't look normal. So the immune system can also trigger those cells to undergo apoptosis. So MHC, it stands for Major Histocompatibility Complex. Again, I'm not expecting you to know all of the details of how this works. That's why we offer a course in immunology. But antigen-presenting cells, including regular nucleated cells and our professional antigen-presenting cells, present antigens to T cells using MHC. So here they're showing you the structural differences between class 1 and class 2. The binding pockets are slightly different sizes, so the antigens presented by class 1 and class 2 will be slightly different sizes. Class 1 MHC, again, remember, will present to those cytotoxic or CD8 positive T cells, whereas MHC class 2 will present to CD4 positive T cells or helper T cells. Class 1 are found on all nucleated cells, whereas class 2 are found only on those professional antigen-presenting cells. And again, here it's showing you the pathway. MHC class 1 is sometimes called the endogenous pathway because it's presenting intracellular antigens. So there's a antigen that's found on the inside of the cell, it's digested, loaded onto the MHC in the ER, and eventually expressed on the surface of the cell. Class 2 MHC, they are sometimes called the exogenous pathway. So again, you have an antigen that's been internalized and phagocytized, digested. It's loaded onto the MHC in various vacuoles that are then exported to the cell surface. So you have presentation on that class 2 MHC. It is possible to have cross presentation where exogenous antigens can also be shown on class 1 MHC, but we generally tend to see, this class 1 or class 2. And so here they're showing the class or the cross presentation, where the phagocytized antigen can be put in the ER and then displayed on the surface of the cell. So this is probably going to be something really common in dendritic cells, which are those professional antigen presenting cells, and also possibly in macrophages. The thing that's important to remember about the antigen-specific immune system is that it takes time during the primary infection for this antigen-specific immune response to occur. So you see the peak is days after the infection has started. So really when we first encounter an infection for the very, very first time, when our immune system is what we call naive, it's never seen that pathogen before, meaning unvaccinated, not previously infected, we're really dependent on our innate immune system to hold it off as long as possible until that adaptive or antigen-specific immune response can kick in. And then you'll see that we maintain a population of T cells that are antigen-specific that remember that infection. And so hopefully, if everything works according to plan, if you are exposed to that same pathogen again, your adaptive immune response, your antigen-specific immune response, will occur much more rapidly. This is showing the T cell responses in CD4 positive T cells. So it depends on which cytokine stimulated the CD4 positive T cell. You'll see we can drive T helper 1 responses, T helper 2 responses. These are called sometimes T helper regulatory or T regulatory T cells, and then Th17 T cells. The thing with immunology, just like medical microbiology, it changes super rapidly. When I took immunology, there was only Th1 and Th2, and now there's a couple more. So again, it depends on the types of cytokines that stimulate these helper T cells. So T helper 1, early local responses, inflammatory, cell-mediated cytotoxicity, type 4, delayed type hypersensitivity. So this is a type of like allergic response. T helper 2, later systemic responses, promotes humoral. So humoral is going to be B cell or antibody responses, and it can help tamp down an inflammatory response. Remember, later we're going to want to tamp down an inflammatory response. T helper 17 activate the epithelial cells, neutrophils, promote inflammation and autoimmune responses inhibited by 1 and 2. T regulatory suppress new responses and regulate autoimmune responses. So all of these T cells, it looks like sometimes they might even like counteract one another, and they do, but they exist in a really important balance in our bodies. And as long as those T cells are found in that appropriate balance, we'll maintain an appropriate immune response, and we'll be able to fight off our infections. When things start to go, and the scientific term I'm going to use is wonky, that's when we begin to have issues with things like allergies. Allergies are an inappropriate immune response. Autoimmune conditions, inappropriate immune response. And even driving that cytokine storm or septic shock that we talked about in innate immunology, those are all inappropriate immune responses. So when these helper T cells are not found in the appropriate balance, that's when we can really start to run into some issues. The other major group of T cells, if we classify them as helper and cytotoxic, are the killer T cells or cytotoxic T cells. The cytotoxic T cells specifically are CD8 positive, and they encourage, as I mentioned before, either cells that are infected or cells that have become cancerous, they've transformed, to undergo apoptosis. So that's a really important mechanism for eliminating viral infection, or for helping to control the development of cancer. So our CD8 positive T cells are critical for that. The other type of killer T cell we talked about under innate immunology, they're the natural killer T cells. They lack CD4 or CD8, so they are activated in a different way. They release pro-inflammatory cytokines, so as more of a general immune response, which is why we talked about them under innate immunology. So they're also very important for our early response to infection, and they are triggered specifically by some of those mycobacterial organisms, like Mycobacterium, that have the mycolic acids, that waxy layer. So again, even though they are a type of T cell, they are more of an innate part of the immune system because they don't respond to specific antigens like CD8 and CD4 positive T cells do.